Okay guys, we'll give this another shot. Won't be there tomorrow, as you know. Sorry about that. I plan on being there Tuesday. We can jump in with Fish ID, start to really get into the diversity of vertebrates. Fish is a good place to get started, actually. Remember we said 30 out of 60,000 species of vertebrates are fish. That's the really big theme in the class, is this diversity. And before we get into the diversity, right, we need to study that process that has created the diversity, and that is evolution and speciation, and finding a way to categorize animals into groups and to study their relatedness, and that's really all this lecture 1 business. We talked a lot about there's mechanisms that actually lead to the change. We talked about mechanisms like natural selection and genetic drift that push populations apart. One, because of differential survival and reproduction, and that is the genotype that is most fit for a environment is the one that will lead to survival and reproduction. And as time goes on, that trait becomes more and more reinforced, I guess you'd say, in that population, and so that population becomes adapted to that environment. Okay. Genetic drift. Why does there change? It's just totally random. A lot of changes actually just random. And then we looked at how those changes can accumulate through time and really culminate in two populations sort of diverging and no longer sharing genes, right? Gene flow is stopped, so that any mutations that happen in this population will not flow to the other population through gene flow. Okay, and so that's kind of where we left off. Let's see. For Pete's sake, stop. Okay, and so this is where we stopped. Sorry, my dogs have joined me. I'm trying to keep them out of here, but this is, recorded history. This is individual people and families. We very rarely know this. This is not really what we're talking about. This is an analogous type of situation where we're talking about we're really normally in zoology and evolution we are trying to piece together what happened in the far past. We're not keeping track of individual people, right? So what we usually do is come up with these trees or phylogenies or cladograms as a way to, and this is how science happens. We come up with a hypothesis. We come up with a testable, possible answer to the question of how these organisms are related. Okay, it's one that is changeable. It's not immutable. It is always on the table. As new evidence comes in, we can change that, and they do change a lot, actually. So a lot of times, if you textbooks, the problem with textbooks is they're always coming out with new editions, and it seems like they're just doing it to charge you more money. But in actuality, if you look at the phylogeny of particularly animals, it has changed a lot in the last 10 years. I don't know so much about vertebrates, but these things do change. We see the branches, the tips of the branches represent species or individual animals in some cases. It's the entity, whatever entity we are measuring. Could be families or genera. The nodes where the splits happen represent that sort of diverging of paths, evolutionary paths, and those would be speciation events. And then we can look at how characters have changed as these splittings, the splits have taken place. Okay, so here's just a given phylogeny or a cladogram, taxon A, B, and C. We see here are the nodes that I was talking about, would be these splits, and here and there there's the branches. Okay, so we don't know when this happened. We can sometimes date these speciation events. We don't know what the organism is that would have been the common ancestor of B and C, but we know that it existed. And then B and C are more closely related because we could say like maybe they're first cousins because here's the grandparent. They share a grandparent. AB and C are all related because they share a great-grandparent. This is not really grandparents and great-grandparents, but just an analogy for you to prove the point. Okay. So we can plot the characters on here too, and I'll show you what I mean by that. Now, when we're looking at characters, right, it's important, and this is really the foundation of this approach to studying evolution called cladistics, and we're going to see how we can use cladistics in this to build phylogenies and then to study the systematics of vertebrates. Okay, so this again, there have been many approaches to phylogenetic systematics, but cladistics has proven probably to be the most useful one. It's the one that people use these days. So for any character, really we're going to focus on a specific type of character, which is a derived character, which means it's relatively recently arisen, and we find it in the common ancestor to the group, right? Whatever clade we're talking about, whatever group we're talking about, it's shared among members of that group, and it's found in the common ancestor, okay? But not before that. So it is found just in that group of organisms, and it's shared among all members of that, okay? So that's called an apomorphic trait, is one that is recently derived and is found only in the common ancestor and in that group, right? And a synapomorphy is a shared derived character. Plesiomorphic characters are not really useful in cladistics. These are ones that are just shared by too many people to really, or too many organisms in that group and the tree to be informative. Okay, so a plesiomorphic character in vertebrates would be the presence of a notochord, right? It's found in all vertebrates, so it's not going to be helpful for us to determine where turtles fall out in the tree of vertebrates. Okay, we're going to be looking for more recent shared derived characters. Okay, so here's an example. This is a group of darters, Ethiostoma, right? Ethiostoma jordani, rubrum, bellum, wapiti, sanguifluum, and vulneratum. Okay, so we see there is a time aspect to these phylogenies. The further down this way we go, the older the group. We see really we have two groups. One includes jordani, rubrum, bellum, and then we have this other group here. A plesiomorphic character would be really the presence of eggs and bearing eggs. So this is the primitive character state. So the ancestor to all of these darters we see in the slide here would have buried their eggs, and then more recently, right, as time marches on, there was an evolutionary change, character change, where the new status is to actually not just simply bury your eggs, but to attach them at the underside of logs or underside of rocks. Okay, and so attaching eggs would be found in this. We can infer, we don't know, but we can infer that the common ancestor of these three species would have attached their eggs, and therefore all of these species also attach their eggs. It's something that was inherited from that common ancestor. Okay. So, plesiomorphic and a apomorphic character state. So, three different taxa. Here's really 4 different possible ways to that they could be related to each other, right? In this phylogeny, B and C are more closely related to each other, and that most ancestral species is A. Here, C is the most ancestral, and A and B are more closely related. And then in the 4th one here, A and C are more closely related and more recently derived. So these would be sister species, and B would be the ancestor to them. Oh no, B would not be the ancestor to them, right? This unknown organism down here would be the ancestor to A and C. I misspoke. Okay. Now, what about this one at the top here? This is what we call a polytomy, right? And what this tells us is we don't know. This is basically acknowledging uncertainty in the evolutionary relatedness of these groups. We need more information, right? Now, so for example, the attaching your eggs, right? The egg placement would be a character. If it's found in a group of taxa, and it's similar among those taxa, and the reason that those taxa are similar is because they all inherited that character state from their common ancestor, then we call that a homologous trait. Okay, so similar by ancestry. Now, it could be that it's not exactly the same, so you could have the same structure, a homologous structure, as an ancestor, or as a related, somebody else in your clade or in your group. You may all share this character, but you use it in a very different way. It's still homologous. Okay, so a great example of this is the forelimb of all vertebrates. Right, they're all homologous. They all have 1 upper arm bone, 2 lower arm bone, 5 digits. Right, whales use them as flippers. Bats use them as wings. So they're morphologically dissimilar in a way, or the way that they function, the way that they're used is dissimilar. But if you look at the bone structure, they are very similar. Feathers of birds and scales of reptiles are homologous structures. The lung of our lungs are homologous to the swim bladder of fish, right? And so all these things are similar because of common ancestry. Okay. So homologous. Right? A frog's limb and a horse's leg are homologous. The same genes that turn on during development to make this bone are the genes shared in all 5 of these organisms. Why do we all have the same genes? That says, okay, during development we're going to make 5 digits because we all are cousins, right? Okay, now, whether something is a derived character, whether it is a shared derived character, it's all relative depending on what groups you're talking about. So, for example, with the forearm of vertebrates, right? Or really, actually, we're talking about tetrapods here. When you're comparing lizards and mammals, this is not this would be more of a plesiomorphic character. This is not a shared derived character among mammals and these reptiles, because the common ancestor had this trait, but so did the previous common ancestor, i.e. the character state is older than the common ancestor of these two groups. Okay. See if I can say that again. Versus if you're considering amphibians, mammals, and these reptiles, then the tetrapod condition would be a synapomorphy. It would be an apomorphic trait, and it would be shared among all members of the group and found in the common ancestor. Okay. So it just depends on how far back we're going on the tree. It depends on how inclusive we are with the groups that we're including. Okay. Whether or not something is synapomorphic. Okay. Now, sometimes you can have similarity by common descent, and other times you can have similarity for a very different reason. Okay, so the characters that have different evolutionary histories but evolve to be similar. So similar character state, but each group that has that character state arrived at them differently. Okay, so we'll look at a couple of examples here. Birds and bats both have wings. Okay, but that's not because the common ancestor of birds and bats had wings, and therefore everyone from there on out had wings. Okay, it's because birds started over here, bats started over there, and then they converged on a similar character state because of maybe similar selective pressures. Parallel evolution is a little bit different in that, right, rather than starting very differently and converging on the same character state, these two groups are both rodents, and so they evolved more or less in parallel in the same type of environment, and similar types of genes, similar developmental pathways led to them being similar in appearance. Now, that said, they are closely related to each other. However, by definition, their common ancestor did not have these long tails and little jumpy feet in the back, jumpy legs in the back. Okay. Sometimes two organisms can look similar because one of the organisms has sort of reverted back to a previous design. So dolphins are a good example of this, right? We know mammals evolved from some type of reptiles, which evolved from some type of amphibian, which evolved from some type of fish. Okay, so dolphins are much more derived than sharks. Sharks are an ancient lineage. Dolphins are mammals, which much more recently appeared, but there was a group of these mammals that actually sort of evolved back to an aquatic existence. And when you go into water, the best shape is similar to sharks and dolphins, right? So sharks and dolphins have the same shape, not because they common ancestor looked like that. Their common ancestor would have been certainly some type of aquatic organism, but dolphins evolved from terrestrial organisms that would have been tetrapod organisms, right? So, yeah. Homologous. Homologous, okay. If you look in early development of all vertebrates, right, they'll have tails at some point. They'll have gill slits, right? Very, very similar at this point in development. Why? These are homologous structures. The common ancestor to all vertebrates had tails. It had gill slits. So why do we have them? Because that's just that's our starting point. Okay, we evolved all of that sort of developmental baggage when we became mammals and became humans. Okay. Now, this similarity here in terms of modification of the forelimb into a wing is certainly not homologous. This would be a good example of a homoplasy. Okay, so we have a eastern bluebird, some type of pterosaur, and a Samoa flying fox. Very, jeez Louise, very differently related organs. We have an archosaur bird, which would have been, left over from the dinosaur lineage. Pterosaurs were not dinosaurs. It was some ancient type of reptile. And then we have a modern mammal. So obviously the ancestor to all of these organisms would have been some type of reptile, and that reptile certainly would not have had wings. So these three lineages independently evolved through natural selection and became flying mammals, or flying animals. Okay. So here we have a very similar character state, right? We have mole. This will be a placental mole, like we have here, hairy-tailed mole, star-nosed mole. We have these all around on campus. If you fly to Australia, you dig a hole, you're going to find this little guy here, marsupial mole, right? Anteaters, numbats. I mean, look at the shape of their body. If you look at their skull, this is just some little generalized small mammal. We have arboreal mammals with prehensile tails. We have mammals that, in both places, that climb trees and have this flap of skin between their fore and hind limbs that allow them to glide. We have the small carnivore. We have the large carnivore. So obviously we see very similar character states between these two groups. And why is that? Is that because of the common ancestor of placental mammals, or placental moles and marsupial moles had small eyes and short pelage? No, in fact, here's the amazing thing. If you think about it, all of these organisms here are more closely related to each other. So the marsupial mole is more closely related to the Tasmanian wolf than it is to the placental mole. Okay? So we saw this adaptive radiation in Australia. The original placental or marsupial mammal in Australia evolved into all these different niches. Okay? So these are all a great example of homoplasious characters. And who cares? So, and again, this is really what this lecture is about. We're faced with all of these diverse species, okay? And we need to try to reconstruct and piece back together. Really, systematics is just a huge puzzle. Is you looking at the evidence, what does it point to in terms of how did we get here? How did we get to these 60,000 species? Who's related to who? What were the changes that took place in what sequence that led to the current community of vertebrates? Okay, now, shared derived characters, these homologous characters, right, will really be helpful in finding good groups. These are what we call monophyletic groups. We'll come back to that, but in cladistics we only consider monophyletic groups. So homologous structures will identify good groups, right? And why are they good? Because they're all related to each other. How do we know they're related to each other? Because they share a common character state. And that common character state was evolved and passed down from the common ancestor. Now, on the other hand, a homoplasious character state, if you put all the vertebrates that have wings into a group, you're going to identify a group where the species are not really closely related to each other, right? And we're going to make these groups that are called non-monophyletic groups, and they are really just the bane of systematic existence, right? So homoplasy really can confuse our ability to reconstruct evolutionary histories. Okay, and I'm going to show you an example from the amphibian world. Here, the branches are not individuals, they're not species, these are genera. So if we go to the creek at Dr. Flint's, we are going to find 2-line salamanders and 3-line salamanders in the genus Eurycia. We're going to find red salamanders in the genus Pseudotriton. Probably none of these other ones we're gonna see. Now, these genera, species in these genera, and all of these too, Amphioma, Necturus, these are mud puppies, Ambystoma would be like spotted salamanders, they do live on campus. All of these groups here, except the ones in the green, have an aquatic larval stage. So they are true amphibiotic animals. They have amphi meaning two, biotic, two lives. They have an aquatic stage and a terrestrial stage. Okay, and certainly the first salamander would have had an aquatic larval stage. In fact, the most ancient types of salamanders are the cryptobranchids, including the Japanese giant salamander, which I showed you guys pictures of. They're fully aquatic. Okay, and it makes sense because they evolved from some type of fish, which are aquatic organisms. Now we do get these very derived groups, Vidracoseps, Aneates, and Plethodon. We have Plethodon on campus, the slimy salamander, those black and white ones that leave a lot of sticky on your hands, are in this genus Plethodon. And then we have green salamander that lives in North Georgia. All of these genera are very derived and different from other salamanders in that they are what we call direct developers, meaning they just skip right over the aquatic larval stage. They go adult, egg, the egg hatches into a juvenile that really looks just like the adult. They don't go through this aquatic larval stage. So the question came up is, where does this other genus called Desmognathus fit in? Desmognathus is a genus I've studied. I studied species Desmognathus welteri for my thesis. It is a stream salamander. It has a 3 to 4 year larval stage. So egg hatches in the water to this little gilled larva, which lives there for 3 or 4 years, and then it transforms into a semi-aquatic adult. A whole bunch of aquatic type species in there. So the question came up, where would that genus fit into this family tree? And certainly nobody would have placed them near this green box, right? Because they have aquatic larval stage. Okay, so same groups. Here's Plethodon. Here is Plethodon whirlii. This is the species that the species group that I had you read the paper about, and we will get to that, discuss that paper, the Plethodon whirlii group, Punctatus and whirlii. A bunch of other species very quickly speciated into this adaptive radiation. All direct developers, totally terrestrial species. Okay, and then we have sister to this genus are the Ensetinas. These live out west, totally terrestrial. And then we have the Aneates, totally terrestrial. And then, lo and behold, right up in the middle of all of these direct developing terrestrial, totally terrestrial salamanders, we get the genus Desmognathus. Okay, so we have aquatic larval stage, aquatic larval stage, aquatic larval stage. Boom. Here we have this ancestor here gave rise to terrestrial organisms, gave rise to terrestrial organisms, and then at some point there was a reversal from terrestrial back to aquatic. Okay, this is very surprising. I mean, I'm getting all excited about this, and I'm sure you guys will get tired of seeing me get excited about this, but think about this. The plethodons and Aeneides, inside of their egg, they basically go through the aquatic stage inside the egg. They have these huge gills. They absorb oxygen through the porous egg membranes, and then they grow very quickly, they develop very quickly, and they jump through that aquatic stage, and then when they hatch, the gills go away, and they're ready to live on land. They never have need for the water. Okay? So, certainly, Desmognathus, the ancestral state, character state, here at this common ancestor would have been this terrestrial existence, right? Because green salamanders and black salamanders don't have aquatic larval stage. But somehow this re-evolved into a group that has aquatic larval stage. It was very, very surprising to amphibian biologists, and this was in the last 20 years that we figured out this. But it gets even more interesting. If we take this part of the tree from Phagnathus up, and we explode that, we put in some more species there, then it becomes even more surprising. Okay, here's the Ensitinas. In fact, we talked about these in General Zoe. This is the ring species, Ensitina escholtzii. Okay, here's the green salamanders, terrestrial. The most, and what would be the oldest group of this, or Desmognathus phagnathus, would be the Phagnathus. This is we can see here. I'm trying to point to the screen here. This is the red hill salamander, described, I think, in the 1980s from just a small part of Alabama. It's the only place you find this group. And it is direct developer. They live in these the black belt of Alabama, on these steep hillsides. They live in these burrows. They lay eggs. The eggs hatch into this amazing little salamander. Now, if we look in the genus Desmognathus, the oldest, most ancestral group here would be the black-bellied salamander. Well, no, actually, back here, the pygmy salamander, Desmognathus rhydi, which is a direct developer. So we see even among the genus, the oldest species are the terrestrial species. And it wasn't until recently in this genus even that we get these species that have these very long larval stages. So very peculiar, and it would fly against all the predictions, right? So here, to kind of go back to our discussion of homologous and homoplasius, the presence of a larval stage among these lungless salamanders turns out is homoplasius, right? Because it pops up here in this group, and then it pops up again up here independently. We assumed that larval stage would have been, aquatic larval stage would have been homologous, and that the desmogs would be down here in this group, but that proved to be very wrong. Okay, so isn't it possible that those, and we just seen, now again, is this the truth? Do we know this to be true? No, this is our hypothesis. This is very strong evidence for this hypothesis now, but we don't know for sure. And then we've seen here that similar character state evolved more than once. Okay, but on average, in the absence of other evidence, right, we are going to go with what we call the most parsimonious explanation for evolution of a group. We are going to settle on the simplest explanation, the one with the fewest assumptions. Trees with fewer numbers of evolutionary changes, right, are more parsimonious, and therefore, all things being equal, until we get more evidence, those are the ones that we're going to pick. Okay. Now, it is science. We know science is not perfect. People that I know, I don't do a lot of phylogenetics. Really, very little at all. The Worley paper is kind of an exception, but I didn't build those phylogenies. I do have friends, and this is what they do, and they have basically told me that almost all phylogenies are wrong. Okay, it is a hypothesis. We don't know them that this is the truth. There's always going to be mistakes in them, right? And so we need somewhere to start. We need something that's testable and that we can make progress on. We can collect new evidence, right? The addition of more taxa and more characters will provide us with more evidence, and therefore our hypothesis will become more and more accurate. Okay? Taxonomy, the way we're grouping organisms, the way we're making sense of 60,000 different vertebrates, it is a science, and it's really an art. And we use hypotheses, and we never know that they're actually true, but it gives us a starting place. Otherwise, right, there's just be total chaos, okay? Be paralyzed with chaos. So, phylogenetic classifications using cladistics. Only recognize on monophyletic groups. These are testable hypotheses, right? It is our best, most accurate reflection of evolutionary history, okay? And it's interesting, they have predictive power, okay? So we can look at extinct species, and based on their character state, we can make a prediction about where they should fit into these phylogenies, right? And also, when you make the phylogeny, it can give you hypothesis to test about whether character states, shared character states, are homologous or homoplazious, right? So we go back to the same group of darters here. At first, we had two polytomies, right? Jordani, rubrum, and bellum were all connected in one, radiating out of one single space. So what that told us is all that we knew was that Jordani, rubrum, and bellum were more closely related to each other, and that they were ancestral to this other clade. Okay, but as time goes on, and as further studies happen, and as really this would be more of like a natural history type of thing, we realize that right, Jordani is the oldest, and then the character state change is to a rounded tail, right? We're now able to differentiate and to piece together the history of this group, okay? And then maybe it goes to this stage where we can now know the sequence of speciation among these three groups, but we still have a polytomy here, right? Continue to study, maybe throw genomics at this, maybe use genetics, maybe study behavior more closely, right? And then eventually we are able to fully resolve the tree and understand, the sequence of, or come up with a good hypothesis about the sequence of this evolution of this group. So, that in a nutshell is about phylogeny. and how these things are put together, how they're useful. You have read this paper that I handed out about the Worley Salamander group, and I think you can see that as an application of making sense of this group. We have this group of salamanders. We don't really know whose species, who are not unique, distinctive species, and who's more related to who, and how this group evolved, and how we used a new technique called genomics to really provide some clarification for how the group is related and how to make sense of the variation in that group. And then, probably more importantly, was how we were able to take that phylogeny that was produced and change the taxonomy of the group. Okay, and that sort of resolves that group and makes it just easier to communicate about and easier to conceptualize the diversity in that group. Yeah, we'll discuss that paper next time. I just want to, because I was involved with the project, I'm able to provide some more details and sort of the backstory about it. So Wednesday we will continue talking about the application of phylogeny and taxonomy and systematics, and really as illustrated through that paper. So we'll see you then. Hopefully I'll see you on Tuesday before that. Thanks.